Peptides and proteins are biopolymers, like we mentioned in our previous lessons, made up of amino acid monomers linked by an amide bond. When they are less than 50 amino acids, we refer to it as a peptide, and when they are more than 50 amino acids, it's called a polypeptide or a protein. Amino acids are classified according to the location of the amino group, where it is attached to the carbon atom, and we can have different positions. For example, in this one, we have alpha amino, meaning it is attached to the first carbon next to the carboxy group. Then beta amino is when it is attached to carbon number 2 in relation to the carboxy group. Then we have gamma amino, which is attached to the third carbon in relation to the carboxy group. And finally, we have delta amino, which is attached to the fourth group in relation to the carboxyl. So that is, we use the relationship of the attachment of the amino group. So generally, the amino acids that exist in nature are alpha amino acids, which we have said the structure has one carbon where we have the attachment of the amino group, and this carbon is what we call the alpha carbon. And this is now what we call the R group side chains that now differentiate one amino acid from the other, because this other structure is the same. So the backbone generally of a protein will therefore consist of the amino group, the alpha carbon that is attached to an R group, and then the carboxyl group, and then again we start with the amino group, and so on and so forth. There are 20 genetically encoded alpha amino acids found in peptides and proteins, and usually they have the name and then an abbreviation of 3 letters and an abbreviation of 1 letter, which you should actually know. You should read them and understand them. So these are the 20 amino acids that we find. And remember, in our previous lesson we talked about essential and non-essential. And essential, we said they must be provided in diet because they're not made in the body, while non-essential are made in the body. So kindly pause your video here and check out the abbreviations of these 20 amino acids. Stereochemistry of amino acids, which refers to the spatial arrangement or configuration. Usually we have 2 configurations: dextro, which means right, on the right side, and we have levo, which means on the left side. So the natural configuration of the alpha carbon is usually the left, such that the amino group is on the left side of the alpha carbon. But now the D conformation would be a mirror image of that, which will mean that the amino group is on the right side, but this is the one that exists in nature. Then acids exist as azotarion, and azotarion is a dipolar ion which has both positive and negative charges, and this will mean that the overall charge is neutral. For example, here we have, remember, our structure of an alpha amino acid. So usually the amino group is going to be positively charged and this will be negatively charged, and this will mean this counter charge, so that it gives it a neutral charge. And this pH, the pH at which this amino acid will exist in this form, is called isoelectric point, Pi. So let's understand this charge better. So amino acids are amphoteric, and that means that they can either act as either an acid or a base. So amino groups usually act as bases, and bases you will accept a proton, and in this case a proton is the hydrogen ion. So pH usually is a scale that runs from 0 to 14. We did this in primary school, so I know most of us are aware of pH. So then 7 is the middle, is usually the neutral, and then towards this side indicates acidity, and towards 14 indicates basicity. So when the pH is below 8, the amino group is going to be charged. This is because at this below where we have acid, we have an increasing concentration of hydrogen ions, and therefore it is going to accept the hydrogen ion so that it is NH3 plus. Now above pH 9, the hydrogen ion concentration has reduced or goes reducing as you go towards 14. This means that the amino group is going to donate hydrogen, and therefore it will be NH2. Carboxylic groups usually act as acids, and this means they donate protons. So when we go to below 2 pH, that means acidity, we have hydrogen ions that have increased in concentration, and in this case, because a carboxyl group has a hydrogen, it means that it will remain, it will not give out more hydrogen, because already hydrogen protons are already a lot. Then above 5, this we are going towards having less hydrogen ions. Now in this case, we have less hydrogen ions in the solution, then the carboxylic group is going to donate a hydrogen ion. So we can review it here. At low pH, that is at acidic condition, proton concentration is high, and therefore amines and carboxylic acids are protonated. That means they have a hydrogen, and in this case this will be charged, and you realize that this will not be charged, but it's protonated. It has the hydrogen, because this one normally is NH2, but now you can see it has an extra hydrogen. Then at high pH, that is basic, proton concentration, that is hydrogen ions, are low. Therefore amines and carboxylic acids are deprotonated. So this goes back to the original state, and now this one will give out its hydrogen. At about neutral pH, amines are protonated, that is around neutral pH, they are protonated, while these are deprotonated. And now here is where we have the Zwitterion condition. You remember we talked about Zwitterion, where it has two charges, both positive and negative. So here is an exercise for you. I give you 1 minute, try figure it out, then we do it together. So this is the chemical structure of glycine. Glycine is the simplest amino acid that has the R group as H. So we said that this is an non-existent form because normally it will exist with charges on this side. So we want to know at pH 1, this is acidic, this is neutral, and this is basic. So at pH 1, that means hydrogen concentration is high. So what will happen to the amino group in this case? It is going to be protonated, and this is also going to be protonated because the concentration of hydrogen is high and it will not lose a hydrogen. At this pH 7, we are saying it is the neutral pH, then both of them are going to be, so this is going to be protonated and this is going to be deprotonated. At pH 12, we are saying that hydrogen concentration is low. So this one will be deprotonated, while the carboxyl group is going to also be deprotonated. So deprotonating this makes it have no charge. Deprotonating this one makes it have a negative charge as it is losing a hydrogen. So let us now classify amino acids according to functional or R group, the properties of the functional or R group. We have what we call aliphatic, aromatic, amino group, hydroxyl group or alcohols, sulfur atom, amide or non-basic nitrogen side chain, acidic side chain, and basic side chain. So we begin with the aliphatic. They're also called non-polar or alkyl group side chain. We already talked about glycine. It's the simplest. It just has a hydrogen as the R group. And remember, we said it is important to remember the 3 abbreviation and the single abbreviation. Alanine has a side chain of a methyl group. Valine, you can see. So these ones, really, you just need to observe and be able to remember them. So these are alkyl groups with different amounts of carbon atoms. Then we have those that have an aromatic group side chain or a ring. We have phenylalanine. It has a 6 carbon ring. Tyrosine also has a 6 carbon ring. And tryptophan has a 6 carbon ring combined with a 5 carbon ring, and this also makes it be called an indole aromatic group. And this one is also called phenolic OH because of this OH here on the ring. You can see that this one doesn't have an OH group on the ring. Then we have amino acid proline. This is a special one that usually adopts a secondary structure called amino group that is a rigid cyclic structure. So this is the amino group there, the alpha carbon, the carboxyl group. So you can see it has joined into the amino group, forming a cyclic structure that is quite rigid, and this is like a secondary structure. We will be looking at structures of proteins, and this is usually a unique amino acid called proline. We have amino acids that are alcohols because they have a hydroxyl group, and we have serine. You can see it has OH. We have threonine, has an OH group, and tyrosine. Remember, tyrosine was also aromatic because it has a ring, and then again it's a phenolic OH because the ring has a OH group. Amino acids that have sulfur, we have cysteine. Here it has a sulfur atom, and here we have methionine. So this one we call it thiol group, and this one we call it sulfide because the sulfur atom is in between carbon atoms. Then we have amides because they have nitrogen. So you can see asparagine has an amide group. We have glutamine also having an amide group. And then we have tryptophan having a group called indole because it has the nitrogen within the ring. You can see that. And it's also aromatic, of course, because these are 2 rings, a 6 carbon ring and a 5 carbon ring. Then we have acidic amino acids, also called non-alpha carboxylic group because they have a carboxylic group but it's not attached to the alpha carbon. Remember, this is the alpha carboxylic group, but now it has another carboxylic group that is not attached to the alpha carbon, and that's why it is non-alpha carboxylic group. Then we have those that are basic. We have lysine, we have arginine, and we have histidine. So this one has epsilon amino group, that is at carbon number 6. 1, 2, 3, 4. Carbon number 5, sorry. This is epsilon. Remember we were naming them alpha, beta, gamma, and delta. Then epsilon. Then we have guanidino group here. That's a guanidino group. And then we have imidazole ring. This is an imidazole ring. It has nitrogen. So those are basic amino acids: lysine, arginine, histidine. Now you have realized, even as we were going through them, that some of them have side chains that have a charge, or they are ionizable. It is important to note them because now, remember we said that amino acids exist as zwitter ions, but now if the side chain has a charge, it means that the property of that amino acid is an overall positive or negative charge. So here is an exercise that you will do, defining a non-alpha amino acid. I've actually mentioned them, but now you can get to describe them, and then the general uses of these non-proteinogenic amino acids, and then discuss the following non-proteinogenic amino acids. Thank you.